Really interesting bone was found in this cave called the Denisova Cave in the Altai Mountains in Siberia. And there were some technical improvements that we came up with in the lab. A guy, Adrian Briggs, a very clever guy, came up with this series of enzymatic steps to basically take this deaminated cytosine and take off the base and then do some other things. So you trim the molecules back to kind of get rid of that error and clean up the data a lot. The data that we got from this Denisova individual is shown here versus the Vindia Neanderthals. Here you see this huge excess of C to T and G to A. It's gone now when we cut off the molecules and we can clean up the data. The bone was really well preserved, so we were able to get a whole lot more data. This first first pass of it, we had about 2-fold coverage of the genome. We now have about 40-fold coverage of this genome. It looks basically like the Vindia Neanderthal in terms of divergence to humans. The distribution of divergence across the genome looks very similar. But when we make a phylogenetic tree, looking at all places where somebody differs in the genome, the Denisova is an outgroup to all of the other Neanderthals. It's a sister group to Neanderthals with respect to humans. So the Denisova is more similar to other Neanderthals than it is to human, but it's more different to the Neanderthal than any two humans who are alive today. So it exists in this kind of intermediate zone where it's hard to say, using terms that we all would recognize and understand, how it relates to Neanderthals. But we can say in genetic terms with great precision how it's related to Neanderthals. We can say exactly where it's different, what % difference is, and make this tree and describe its relationship in this way. But whether you call this a species or a race or a subspecies or whatever is just an endless argument about what those words mean. And in the end, they don't mean anything because no one can really come to a definition that we all agree on. And words that aren't defined in a way we can all agree on don't mean anything. So here's how they're related. They're related as depicted in this tree. Okay, when we did this admixture test, we sequenced a whole bunch more genomes because it was getting cheaper and faster to do that. And when we did this pairwise test to ask, is Denisova more closely related to one individual versus the other? Instead of seeing this bipartite difference between Africans and non-Africans, there was also a bipartite difference, but it was between individuals who live in Papua New Guinea versus everybody else in the world. People who their ancestry is Papua New Guinean have about 5 to 8% of their genome that is derived from this Denisovan individual compared to everyone else in the world. And that is very strange because the bone comes from about right here. That's where the bone was found. So this is a really perplexing thing that remains perplexing even to this day. We have since gone back, and David Reich and Mark Stoneking and some others got more population data from individuals across Oceania. And there's an interesting signal. Here there's a geological and biogeography feature called the Wallace Line that you may have heard of in this part of the world. Pretty much to the east of this Wallace Line, we see lots of Denisova ancestry, and to the west of it, and in mainland Asia, you don't see any of this Denisova ancestry. Lots and lots of things change about the plants and animals that live east and west of this line, including Denisova ancestry and our species for whatever reason. So and here, of course, is the Denisova cave up here. The best model that has been put forth, and admittedly it's a crappy model, is that early on, when humans migrated out of Africa, they came across the Neanderthals and had some admixture, but this is the proposal, one of the first things that they did was stay along the coast and follow the southern route into Oceania. At that time, it was not such a hard thing to do because a lot of these islands were actually connected, and at one time in the past, you could walk from mainland Asia to Australia, not that long ago in the past. So There's some archaeological evidence for this, that humans made it to Australia and this region of the world a long time ago. It was one of the first places that they went when they left Africa. So under this model, the Denisova group, which we have only one representative of, this bone that we found in this cave, actually had a range, it's proposed, that is much greater than just that cave. That there was a larger population that perhaps extended further to the south. And these early, fast-moving migrants were early and fast enough to meet the Denisovans and admix with them, and brought their DNA to where it is found in people today. Later, a slower migration, a northern route, filled in mainland Asia, and we have a pretty good idea that over the last 4,000 years or so has started to move through Indonesia, and this Austronesian expansion move out this way. So that is, if it feels like it's a just-so story, it's because it is a just-so story to explain this observation. But if you have a better model, I would love to discuss it, because this one, it clearly is built around this observation. And there are lots of other interesting, more fantastic potential explanations for this that we would love to talk about. So in the last few minutes here, I'll finish up describing some more data that we've continued to improve the library preparation methods. This is a schematic that shows that we can capture single-stranded DNA. It turns out that lots of DNA from bones is not even double-stranded. It's single-stranded, and all of the ways that we've been trying to get it in the past required that DNA be double-stranded. If you make a method that gets single-stranded DNA too, you can sometimes get 10 times or 100 times more DNA out of these bones. So we did that, got a lot more data, and now for this Denisova, have something like 40-fold coverage of every position in the genome. So we can be reasonably sure what exactly the base was when this Denisova individual was alive, because every position we get to see on average 40 times. So the sporadic error, we see that that's the error base, and 39 times it's something else. So we can put it together with much more accuracy. When we do that, instead of this long branch for the ancient sample, we actually see that the Denisova has a shorter branch compared to humans. And this is kind of a profound thing. We have always hypothesized, we can compare humans and mice and chimpanzees and dogs and see that these branch lengths are somewhat proportional to the time ago in the past that we think the common ancestor lived. But we've never actually seen molecular evolution in action. And here we see it in action by evidence by its absence. This individual died some time ago in the past, and these sequence changes, these mutations that are happening every generation that we can't stop. The machinery that our body uses to copy DNA is very, very good, but it's not perfect. It always makes mutations. Well, this Denisova individual, it stopped doing that some time ago in the past because it died. And all of these generations of mutations that have accumulated in us have not accumulated in the Denisova. It actually has a shorter branch. It has fewer mutations leading to that Denisova than we see in all humans today. And we can use that absence of mutations to ask this question, how old is this bone? How much evolution is it missing? And it turns out that it's missing what would accumulate in somewhere between 74 and 82,000 years. Which is a useful thing because the bone was very small and there's not enough of it to do radiocarbon dating, and radiocarbon dating is not good at that time depth anyway. So we can get some estimate of how old this bone is just by looking at the DNA sequence and asking how much evolution is missing in the bone. We can also do interesting things. I told you we have this different trajectory back in time based on recombination. You're related to your neighbor maybe 10 generations ago at one place and 100 generations ago in another place. Well, there's a really cool method that was developed by Hang Lee and Richard Durbin that uses the fact that when you sequence an individual's genome, you're actually sequencing two genomes. You're sequencing the genome that their mother gave them and their father gave them. So you can compare at every place across the genome, how different was this individual's mother from this individual's father? How long ago in the past did that common ancestor live? And then you can go to the next place in the genome and say, how long ago in the past did that common ancestor live? And if you scan across the genome, there are tens of thousands of independent evolutionary draws that you're getting from the genome. And if the mother and father are two random people, if this individual is not inbred, you can ask, where in the past do I see lots of common ancestors? Is it 3,000 generations ago, or is it 10,000 generations ago? Is there any place where I see lots of common ancestors? Where you see lots of common ancestors is typical of a bottleneck, where the population size would have gone down. So you can put together the broad strokes of how big of a population we came through time by just sequencing a single individual's genome and analyzing it by this method. So the method, the output of it is this time by the population size, and each one of these lines is a single individual, this red one being the Denisova individual. And all of these other individuals, you can see kind of this bottleneck that was about the time of humans becoming a distinct species. And then there is this out of Africa bottleneck that is bigger in the out of Africa individuals versus the African individuals. You can see this from a single individual's genome. And what's interesting, the Denisova, it doesn't have a history beyond somewhere around 50,000 years ago. And for a long, long time, it came from a population that was very, very small. When we saw this, someone made the very astute comment that we shouldn't be surprised that it came from a small population, given that it went extinct. That it's not that surprising. That population size eventually went to 0. So what this tells us, though, is that it wasn't a catastrophe. It wasn't a dramatic event that happened to a population that was otherwise healthy and large and flourishing. This population was barely hanging on for a long time in Pleistocene, Ice Age, Siberia. There just weren't that many of these individuals. It came from a small population that had very little genetic diversity. Okay, so I am out of time, so I'll just tell you a little bit about what we're doing these days. The Neanderthals, we know, gave genes into the modern human gene pool, and my student Brandon Rice is really looking hard at the biological implication of that. Did they give us any genes that are useful for anything, that may have been positively selected in the recipients? And Neanderthals are also useful for probing this time period in evolution where we started being different from Neanderthals, when we had a different trajectory, when this population split happened. We know that this is a very momentous time in our own evolution. We started doing things that we had never done before and that Neanderthals never did. We had lots and lots of symbolic abstract art. We colonized regions of the world that we had never been to before. We got in boats and sailed farther than you can see that there is land with your eyes, which we had never done before. These kinds of things give us evidence, perhaps, that our mind was a little bit different than Neanderthals. And having the Neanderthal and contrasting with humans, we hope to find, perhaps, what the genetic basis of those differences will be. So we found an interesting immunity gene that clearly came from Neanderthals and is selected in humans. We also have found a lot of genes whose variants look like this. They're very high frequency outside of Africa and low frequency or absent within Africa. This we think is from some back migration, its presence here in East Africa. But these genes, this signal is a very rare thing to see. Generally, any variation that exists outside of Africa also exists within Africa. It's very rare to see something that only is present outside of Africa. A lot of it is explained, when you do see that, by Neanderthal introgression, and that is generally at very, very low frequency, that when people have something that came from Neanderthals, very few people have it. They have their own rare thing, and everybody has their own rare thing, but there are a few places in the genome like this where lots and lots of people have something. And this stands out because, against the backdrop of being rare, because it's so common now, it must have been pulled up by positive selection, conferred some selective advantage. And we're working to figure out exactly how to piece that together. I'm going long on time, so I'll just I'll skip through all of that and finish up there. And if you're ever in Santa Cruz, look me up. Thank you for your attention.